So look, I really apologise that there's some very eminent microbiologists in the audience, so this is going to be very, very low level for you guys. I do apologise. But I guess it's, just trying to get everybody on the same page. So we sort of had a bit of basics of genomics, a bit of basics of infection control, and I guess my job is to try and squeeze everything to do with microbiology into 20 minutes, which obviously I won't be able to do. But I think it's really good just to get some terms clarified for everybody, so we sort of have a vague idea of what is an MRSA or a VRE when we talk about these things. And I think also making the point that, again, in the way that genomics doesn't make sense when it's abstracted from infection control, day-to-day work, it also doesn't make any sense if it's abstracted from what's happening in the day-to-day lab and what we do on the bench and our sort of traditional workflows. Because, in fact, if you're not identifying these organisms or you understand how these organisms are identified in the lab, then the genomics is really kind of irrelevant if you're not finding them in the first place. I guess the whole purpose of this project, again, as we've sort of highlighted several times now, is the importance of hospital-acquired infections and preventing these infections. And Crispin's made a nice case for why we should really care about these things. And they are actually unbelievably common. Thousands of patients in Australian hospitals get these infections every year. And, and patients die from these from time to time. It's not a trivial event. And particularly now with multi-resistant organisms, things that potentially might have been relatively easy to treat are now much harder to treat and are associated with worse outcome. So there's figures from various parts of the world. Europe has quite good data on this kind of thing, but about somewhere between a 1 in 10 or a 1 in 20 chance of a patient acquiring hospital-acquired infection while they're in a hospital stay. In Australia, again, it's about 1 in 10 patients will get an HAI at any one time. And it's a huge burden in terms of cost to the healthcare system and, of course, the human cost and delays to surgery, prolonged admissions, additional antibiotics, etc., etc., not to mention pain, discomfort, and general misery. So these are something we really should be caring about and something we should take very seriously. And I'm going to primarily talk about, there's a whole load of potential hospital-acquired infections, but the ones we probably worry about the most are bacterial infections, and these are often potentially the most severe. And I guess one that's been a very clear focus for our genomics project. So microbiology is, it's an interesting field because we're actually dealing with an enormous diversity of life. If you think about the tree of life, at least a good section of that on the left-hand side is all within our wheelhouse of microbiology, which includes millions and millions of different organisms, some of which are not human pathogens and not necessarily particularly relevant to medicine, but we sometimes grow them in the laboratory, where some of them are highly pathogenic and extremely important from our work and understanding how these are infecting patients. So a lot of what we do is really trying to sort of categorize these organisms. We know there's something there. We have to quite quickly work out, what species is this, what strain is this, what antibiotic resistance profile does this have, and how does this inform what we do from a clinical point of view. And historically, microbiology has sort of been doing the same thing for really about 100 years with improvements and tweaking and better technology and better systems. But the basic fundamentals of microbiology have actually been fairly static in terms of really mainly looking at the phenotype of an organism. So what does it look like on a plate? What does it smell like? We spend a lot of time kind of wafting plates in our directions like wine tasters. We look at things under a microscope. So what do they look? What do the cells look like? How are they behaving? Are they moving around? Are they growing in a certain way? Are they like certain growth requirements? And things like obviously resistance patterns to antibiotics, which we test in the lab. And this is sort of traditional phenotypic microbiology that we do, that all labs do all over the world every day, and is the mainstay of our work. But increasingly, obviously, that we understand that phenotype is determined by the genotype, is determined by the DNA that is within that organism. And more we can explore that genome to try and help understand a little more about those organisms. And, for many years, we've had, say, single-cell gene targets. We may have a PCR that will look for, say, a particular resistance gene. We may look at certain fragments of the genome that help us, say, characterize the species of an organism, like 16S sequencing. But more and more we're using whole genome sequencing, where you can take the entire length of that genome, sequence it, and then infer whatever information you need from there. So this is a kind of very typical microbiology laboratory workflow. So we would have some kind of culture step. This is a blood culture system, so this is obviously the most important thing we probably do day to day. A patient comes in with suspected sepsis. We take a volume of blood and we grow it, looking for bacterial or fungal organisms, say, growing in their blood as a cause of their clinical presentation. And then we grow something over a period of time. This may take a day or two to come through the system. We put it on, say, some kind of media like an agar plate, and we look and see that, oh, that looks like a kind of Klebsiella or something growing. And then we will do some kind of further testing. Now, historically, there used to be a whole batch of biochemical tests or bench tests that we would do to kind of categorize these things. We're getting rather lazy in microbiology these days. We just now put it on these mass spectrometry machines, which give us an answer very quickly and very cheaply. So in fact, getting a species identification has become a pretty quick and easy thing to do for the vast majority of our organisms. And this has really revolutionized what we do from a technology point of view. But susceptibility testing is still a fairly slow and rather tedious art with lots of caveats and complexities. We have a whole variety of different ways we can do this. There are some automated systems, which I guess is the mainstay of what we do in Queensland, at least, particularly this instrument called the Vitek. And this is very useful in terms of high throughput result generation. Sometimes we may want to go and do some more detailed testing, so this thing called the MIC, or the minimum inhibitory concentration, looking at the amount of drug required to inhibit the growth of an organism. Or sometimes we may use a slightly more simple, cheap method like disc diffusion, where you have different antibiotic discs that contain a concentration of antibiotic that diffuses through the agar, and you measure that zone of inhibition. That gives you an idea of how resistant these organisms are. And we will talk from time to time, I guess, about MICs, and these are very important. Traditional MICs are determined by doubling dilutions of antibiotic historically in test tubes, and you essentially look for the lowest concentration of an antibiotic that will inhibit the growth of that organism. So if you look here, for instance, you set up your test tubes with dilutions of antibiotics going from very low to very high, and then you can see here you've got the first test tube that starts to inhibit growth is at, say, 64, so that would be your MIC for that organism. And then that's interpreted against certain standards. There's like European standards or American standards. I want to tell you for that organism, with that MIC, that's sensitive or that's resistant. And there's a lot of argument about where those standards are set and how they're done. So in fact, what we do in microbiology, as I said, hasn't changed for a long period of time. So this is probably the most useful test we do in microbiology, which is a very old test, but incredibly useful test, a rather sort of serendipitous discovery by Christian Graham, who was playing around, I think, with dyes at the time. There was a lot of interest in how dyes might be used to treat bacteria. And he found this sort of fundamental discovery that, in fact, there are whole categories of organisms that stain differently under different staining conditions. Essentially, these organisms are now called gram-positive, that take up the gram stain, and gram-negatives that don't stain up the gram stain. And this actually reflects the fundamental differences in the cell wall of these organisms, and actually reflects a sort of fundamental biological difference. So we still use this every day to divide, that diversity of bacteria into gram-positives and gram-negatives. It's a very useful first step in what we do. And then from there, there's usually lots of other potential subdivisions based on other molecular or biochemical tests to try and categorize these organisms into other genus or species groups. Now, the most important thing that we probably see every day in causing severe infections, bloodstream infections, skin, soft tissue is Staphylococcus aureus. And we'll talk a little bit today about MRSA, which is a particular resistant type of Staph aureus. But it's important to realize that this is a really serious pathogen. It causes, one of the greatest sort of, I guess if you think about calling out blood cultures every day, the thing we see more and more causing the most serious disease is often Staph aureus. And it's ubiquitous in the community, and it's also a very common cause of hospital-acquired infections, and it has numerous clinical manifestations. And unfortunately, Staph aureus is a very wily organism, having been very susceptible to penicillin. When penicillin was first discovered, within a very short period of time, penicillin-resistant strains were described, and inevitably over the years, increasing resistance has been recognized in this organism. To the extent now that, the biggest concern has been the emergence of this MRSA form of the organism. So this is methicillin-resistant Staphylococcus aureus. So this emerged in the 60s, but has become really widespread in the last few decades. And we'll talk a bit more about the sort of epidemiology of that. But this is an organism that has really become a problem in hospitals for a long period of time, although interestingly has been declining to a certain extent in recent years. And when we think about hospital-acquired infections, MRSA historically have been one of the biggest things that we worried about. This methicillin resistance is mediated by the acquisition of a gene called MECA, which essentially makes the organism unable to or makes the antibiotics unable to bind to the organism, so they're not effective. And there are various particular hospital-adapted clones that have become dominant in Australia. So particularly the sequence type 239 clone that's been established in many hospitals. There's a UK MRSA15 clone that has been introduced from the UK in the 90s that also became very prevalent. And these cause, or historically have caused, as I said, a very large proportion of hospital-acquired infections. And if you get a bloodstream infection from an MRSA, you actually have a really high risk of poor outcome and even death. But these are hospital strains that have been circulating and have been emerging over the years, but we've increasingly seen these community-acquired strains, or strains that originated within the community but increasingly the epidemiology has become more and more complex. So you have community strains that are then sort of coming into the hospitals and spreading within the hospitals. So this division between community and hospital strains gets more and more difficult to discern just purely from the epidemiology or even from the susceptibility profiles. And that's why understanding a bit more of the molecular epidemiology is really helpful. And we certainly have we have good sort of molecular surveillance programs underway in Australia that keep track of how these clones are spreading around the country. And there are particular sequence types that are predominant in certain parts of the country. These community strains are interesting because they, although they may be slightly more virulent and they can carry particular toxins that present with more severe clinical manifestation, they tend to be a little less resistant to antibiotics. They still carry that MECA gene, but they don't necessarily have all the other antibiotic resistance genes that mediate resistance to a larger variety of antibiotics. And sometimes when you see this sort of nomenclature here, people will talk about, say, an ST93 MRSA. So this is the multi-locus sequence type. Again, this is just a handy way, a sort of shorthand way of communicating between different laboratories to say, like, I've sequenced a series of genes, I've generated a code that ascribes this sequence type in an international database. And that's a sort of a universal language that we can all communicate between laboratories. Say, this ST93 is the same as your ST93. And then we often talk about this thing called the SCC-MEC. So that MEC-A gene that mediates resistance to methicillin sits within the staphylococcal cassette. So this is like a mobile packet of DNA that carries that resistance genes. Sometimes these are small and may have only carry MEC-A. They may be larger and carry a greater variety of resistance genes or toxins. And these can be typed. And very often we'll see that number at the end of the sequence type, which really just tells you that the SCC-MEC typing is described. So VRE is another important one, which we'll, I'm sure, talk about today. Enterococci are sort of difficult pathogens, and they're intrinsically resistant to a number of antibiotics. They're relatively low-level pathogens. They're very often colonizing the gastrointestinal tract, and often, say, the urinary tract or the skin or a wound. And about 1 in 10 of those may go on to cause a significant infection. So we detect these quite commonly. They may be sitting in the gastrointestinal tract, but severe disease is less common. But they can become very resistant to antibiotics, particularly resistance to vancomycin, which has been the sort of mainstay of treatment of these organisms when they cause an infection. Once they become vancomycin resistant, then they're extremely difficult to treat. So if you have, say, a VRE endocarditis, they're almost getting into kind of untreatable territory. And these are particularly challenging from an infection control point of view. They're very persistent, not only in your human gastrointestinal tract. They can contaminate the environment. They can get into surfaces and survive there for a long period of time. They can be very hard to get rid of. And unfortunately, as you can see, Australia seems to lead the world in terms of prevalence of VRE. So we are pretty good at many things, but unfortunately for VRE, it's a particular Australian problem. And the resistance of VRE is interesting. So it's mediated primarily by these VAN genes, particularly VAN A and VAN B. And you'll often hear people talking about VAN A types or VAN B types VRE. And these are genes that are acquired by these organisms that mediate resistance to vancomycin, but they have slightly different susceptibility profiles. So if you have VAN A, you're usually resistant to both vancomycin and Carbapenem. And these were relatively uncommon in Australia over a period of time, but have now unfortunately become increasingly recognised and replacing many of our more traditional VAN B types. The VAN Bs had a sort of get-out clause in that they were usually susceptible to Carbapenem, so they usually had some treatment options available. And VAN B is interesting because it's probably actually often acquired from anaerobes sitting within our gut. So the infection control implications of this is very difficult because in fact it's probably sensitive Enterococcus faecium that are transmitted in hospital, and then they may independently acquire those resistance genes probably from antibiotic exposure, in your gastrointestinal tract. So we may often be actually missing the things that are spreading, and the more genomics we do, the more we sort of realise what's actually going on is even more complex than we thought, because just tracking the resistance only gives you part of the story. So gram-negative bacilli, again, this is a really important group of organisms that we'll talk about today. So there's particularly the Enterobacteriaceae. They've been handily renamed as Enterobacteriales just to confuse everybody. But these are things that live in our guts, so like E. coli, Salmonella, Klebsiella, and cause a very high proportion of gram-negative infections, so particularly urinary tract infections, bacteremia, intra-abdominal infections. And then there are another group of organisms that we often lump together as what are called non-fermenters, and these are more sort of environmental, opportunistic pathogens like Pseudomonas or Acinetobacter. You may find these in a puddle or in a shower head, but they can certainly cause significant infections, particularly if you're immunocompromised or you have lines put in you or you have underlying chronic comorbidities. And when we think about hospital-acquired infections, if you have, say, a catheter-related urinary tract infection or a ventilator-associated pneumonia, these are the kind of organisms that really start to plague your patients. And gram-negatives are a real nightmare from an antibiotic resistance point of view because they have so many mechanisms available to them to resist pretty much whatever antibiotic you want to throw at them. So they can break down your antibiotics, they can produce enzymes that destroy your antibiotics, so particularly things called beta-lactamases, which we'll talk about, I'm sure, today. They can change the target so the antibiotic can't actually bind to where it needs to go to act. They can modify the antibiotic in some way so it becomes less effective. They can pump the antibiotic out of the cell. They can prevent it from getting into where it's supposed to act. They can change the lipid membrane. Pretty much anything you can think of, as soon as you develop an antibiotic, they've already developed some natural defense against the action of these drugs. So they're very smart and they're very adaptable. And I guess probably the most important thing to consider when we think about gram-negative resistance are these beta-lactamases. So these are enzymes that are produced by these organisms that essentially break down our most important antibiotics, the beta-lactams. So this includes things like penicillin, cephalosporins, carbapenems, our real workhorse antibiotics that most patients get when they acquire infections. And these enzymes are able to essentially neutralize the action of those antibiotics. But the problem with beta-lactams is that beta-lactamases is an absolute schmozzle when it comes to the science. There are thousands of different beta-lactamases described. The nomenclature is all over the shop. So they basically, over the years, have just sort of made up slightly random names based on whatever someone was thinking at the time. So the name of the first patient that was described, where it was first, first found, in location, etc., etc. So none of it has any real sort of logic behind it often. And there are now thousands and thousands of different variants, all of these types of beta-lactamases, and there are more and more described every year. The curation of these genes, or the actual true significance of a lot of these genes, is poorly described. We quite often detect this in genomics, so we'll find there's an OXA 548 or something, and you look it up, and it's just been described in the metagenome of a deep-sea fish or something. You'd have no idea what it means, and no one's actually done yet all the work to characterize the clinical significance of these things. So, unfortunately, we can often detect a lot of things that we don't always necessarily know what they mean. Things are even more complicated in bacteria because a lot of these resistance genes are carried on these mobile packets of DNA called plasmids, and these are a real bugbear from an antibiotic resistance point of view, and certainly one of the reasons why gram-negatives are so successful and have become such a problem when it comes to treating these. Essentially, these genes can be carried on these little circular packets of DNA. Very often there may be multiple antibiotic resistance genes on a single plasmid. And if you can imagine, that creates extra problems, because if you expose that bacteria to an antibiotic, you may not just be selecting for the antibiotic you choose, but the other antibiotic resistance genes that are carried on that plasmid. So you could be thinking, I'm giving someone Bactrim, nice, sort of low impact antibiotic. But if that plasmid has a Bactrim resistance gene and a carbapenemase sitting on it, you're probably co-selecting for the carbapenemase as well. So nothing is simple in this life. And the problem is that all those resistance genes can be transmitted together. So these bacteria can have a kind of primitive form of sex. Very often there may be signals sent out, so you expose your population of bacteria to an antibiotic. Those susceptible strains may send out signals to say, help us guys, and they're sort of good communists, they spread their genes around. They will actually be able to insert some of the plasmid and its associated resistance genes into, say, a susceptible strain, and that susceptible strain will then become resistant, and then all its daughter cells will then carry that resistance gene. So not only have you got this sort of what's called horizontal gene spread, so it's transmitted across the lineages, so you may have different sequence types, say, of E. coli, but it can even transmit across species, so it can go from an E. coli to a Klebsiella to a Pseudomonas. And as you can see, piecing that together is incredibly difficult. So you're not only having to understand the evolution of the sort of bacterial clones, you have to understand the horizontal transfer of these mobile genetic elements. And that's where things get really interesting and really complicated. And you'll sometimes hear us talk about there are different ways of typing these plasmids, particularly these incompatibility types. So we may talk about INC-F or INC-HI2. Again, it gets very complicated, but if you hear those terms, that's what we're talking about. I won't labour this too much, but a lot of what we do in the lab from an infection control point of view is screening specimens. These are difficult because, let's say you take a bum swab, you take a rectal swab, there's going to be thousands of different types of potentially bacteria in there. You want to only really look for the organisms that you're most worried about. So let's say you're targeting ESBLs, extended spectrum beta-lactamase producers, or a Carbapenem-resistant organism. We very often use special media that will only grow those kind of organisms, and also they frequently will have some kind of indicator in the media that will tell us, okay, this is resistant, and this is looking like a Klebsiella, or this is looking like an E. coli. So it really helps the laboratory picking the right organisms in a short period of time. Otherwise, you end up working up loads of different organisms that may not be relevant, and your whole process will be very inefficient. So these sort of chromogenic media you'll often hear talked about, and they have many advantages in terms of speeding up what we do in the lab. But they have differences in terms of sensitivity and specificity. And lastly, I'll just put this in there for the perplexed and the confused. The terminology in this area is a bit of a nightmare. You'll have sometimes people talk about CRE or CPE or CPOs or MROs, and they're all overlapping terms in many ways. The point being that an organism I guess one of the organisms we worry most about are those that resist carbapenems. So carbapenems are like our kind of last line antibiotic that we really want to reserve for our sickest patients. If you have lots of carbapenem-resistant gram-negative organisms in your hospital, you're going to be really challenged in terms of treatment and choosing the right empirical therapy, say, for sepsis. So understanding how these organisms become resistant to carbapenems is very challenging, and there can be multiple mechanisms. The ones we worry about the most are these carbapenemases. So these are these acquired beta-lactamases that may come on a plasmid and make, say, a sensitive strain highly resistant. But equally, there may be intrinsic mechanisms with the organism that can switch on efflux pumps or pour-in changes that will make that able to resist carbapenems. But that hasn't acquired a resistance gene. It's just part of the way that organism responds to antibiotics. So it can be carbapenem resistant through multiple different mechanisms, and those different mechanisms will have different significance in terms of, say, an infection control response. So a lot of what we do is trying to communicate to the infection control teams, yeah, this is a carbapenem-resistant organism, but this is something you really need to worry about. This one, probably less so. So it's stratifying that sort of infection control risk so you know what to do about it. And a lot of the time is trying to identify these new acquired carbapenemase genes that can transmit easily on these plasmids between different organisms and can cause outbreaks. And these are the kind of things that we worry about the most. And there are many different ways you can try and detect these things. This is, I guess, a very brief summary of what we do here in Queensland, and I guess many other labs do a similar thing. But it is really worth, if you're working in infection control, to have a good idea of how your lab does these things. And there are some subtleties and there are some complexities and pitfalls, and things sometimes slip through the net. You know, as I said, there are so many different new types of beta-lactamases around, new genes being introduced. No system is 100% perfect unless you're going to do, say, genomics on every single strain that you grow in the lab. And we have to be selective to a certain degree just because of practicalities. But as a broad way to think about this is you'll be using some kind of simple automated or high throughput system which detects resistance. We use the Vitek 2, which is a very commonly used system. If it flags up with any meropenem resistance, which is a carbapenem drug, we then go on to do some further testing to see, is there a carbapenemase being produced? So we particularly use this thing called the Carba-NP or the beta-carba test. It's a very simple bench test. It takes a short period of time to do. Within an hour or two, you'll know, is this organism producing a carbapenemase? Is it producing an enzyme that can break down an antibiotic? In fact, all it has in that well is an antibiotic with a pH indicator. When you break down the antibiotic, it changes the pH, so the colour of the well changes. It's brilliantly simple and actually pretty good at most carbapenemases. But like everything in life, there's some it misses. So if it's a weaker carbapenemase, it may not go positive, particularly these ones called OXA48s, which we'll talk about a little later today. So we often have to do multiple things. We may put it on a chromogenic plate to try and select out for certain carbapenemases. We may do the Carba-NP test, and we may double-check the MIC of that organism to a carbapenem drug. And it's looking at the pattern of those results will tell us, okay, we're really worried this is a carbapenemase, or actually this is very unlikely to be a carbapenemase producer. And then finally we will do some kind of multiplex PCR, so this is amplifying target genes. In particular, there's 5 carbapenemase genes we worry about that are most common across the world, and that's a simple test that we can do in the laboratory. And what we're going to talk about for the rest of the day is, when you have a high degree of suspicion of a multi-resistant organism, then you can do your whole genome sequencing, and that will tell you potentially where it's come from and how it's related to other organisms that you've sequenced previously.